the outer coat layer, which basically forms these cage-like structures. So what we work on in my lab is to, our aim is to understand how these two layers of the coat assemble and how the assembly is regulated in different conditions. And in order to achieve that, we use a cryotomography-based approach. So we, one of the approaches that we use is to reconstitute this, what we call the budding reaction in vitro, and then to image it by cryo-electron tomography. So we purify all of the COP2 proteins, and we incubate with a membrane source. What I'm going to show you today is done with giant unilamellar vesicles, so totally artificial membranes. And then in the test tube, the budding reaction occur, and then we can image them. This is a slice through a typical tomogram where we can see coated tubules and coated vesicles. And then we use subtomogram averaging to get to the structure of the coat. So in this slide, I'm showing you some of the reconstructed tomograms. So just quickly mention that we have recently started to use ISONET to process our tomograms. So this is a program that uses AI methodology to fill in the missing wedge at low resolutions. And that's really helpful if you have membrane containing systems. It's really helpful to fill the membranes on top and bottom of the tomogram, which I'm sure many of you know that if you have a membrane, usually in the reconstruction, you can't see the top and the bottom, which when you're picking your particles and things like that can be a problem. So if you are having a project like that, I strongly recommend using ISONET. And then here you can see like the raw data, which has just been filtered and denoised, where you can see the two layers of the coat. So basically, we have used this type of data to get the structure of the inner and the outer coat by subtomogram averaging, and that is summarized here. So this is data that we've published, and basically we have the structure of the inner coat here in green, blue, and yellow. This is somewhere between 4.5 and 5 angstrom resolution with crystal structures fitted in it. And then the structure of the outer coat, this is somewhere between 11 and 15 angstrom. Resolution is quite variable. And then we have both structure mapped back onto one of the tubes that we used. And so this kind of study taught us a lot about how the subunits interact with each other, both at the level of the inner coat, at the level of the outer coat, and between the two coat layers, showing us the various nodes of a very complex network. And this also allowed us then to go back to the bench and do mutants and really test what the role of these various interactions is in membrane budding, in determination of the vesicle shape and size. So I'm not going to discuss that at all today. What I want to do is I want to focus on the pipeline that we have been using in the past and how we improved it recently. So basically, this summarizes the main steps that I think most of us doing cryotomography and subtomogram averaging go through. So of course, we need to start with collecting the data, and I don't have anything to say there apart from the fact that all our data that I'm going to show you today is being collected at EMBL through the iNext program. So a big thank you to Wim and his team for really excellent data. And then after we got our tilt series, which is normally nowadays collected with a dose symmetric scheme, we need to go through some pre-processing steps. So these will include motion correction, CTF estimation. In the case of tomograms, you also need to create a stack. And I think most labs doing subtomogram averaging, they have their own in-house script to do all of these steps. And there are a few things that, is this still working? I feel, yeah, okay. There are a few things that we need to keep track of. For example, we need to make sure that we keep track of the order of the data collection to know what the dose that each image has received. And we also, when we do the CTF estimation, for example, we need to make sure we know. Yes, okay. Right. So then we need to align the tilt series. And again, here we have a number of possible softwares we can use. One of the most common is iMod, but there are a number of new software. We normally use Dynamo for aligning the tilt series. And there are also a number of question marks that people are still wondering, is it better to have fiducials or not? Is it better to just do fast automatic alignments, or do we want to pick and select and perfectly center every single gold bead? And one of the questions that I don't think has been really answered yet is, does it actually matter if then when we're doing subtomogram averaging, we are refining the frames with like Tomo CPR or frame refinement, depending on the software. Then we need to reconstruct the tomogram. And this is really kind of not a hard thing, but one of the pitfalls that we found at this stage is that different software reconstruct tomograms in different XYZ coordinates. So this may become a problem if you're picking particles from a certain reconstruction and then you're using a different software to actually process your particles. And yeah, then we need to pick the particles, which is probably going to be, in any attempt of automation of the tomography pipeline, I think that's probably going to be the bottleneck. Probably AI is going to come to aid, but by definition, subtomogram averaging projects are not single particle projects. So the particle picking is really project dependent and is not something that, at least for in our hands, template matching hasn't really worked. So we always have to find the good way to pick a particular particle sort of as in a project dependent manner. So after all of this, you have the subtomogram averaging and classification, which are pretty much streamlined in many software. So I'm going to show you how we use RELION 4 recently to do this. And basically in RELION 4, once you have your particle coordinates and your aligned tilt series, it's all, you know, press the button and it works. So that's easy. But for the first part, as I said, labs have their own scripts to do all of this, where you need to, you know, there's a number of little things that to take into account. They're really not hard, but if you get them wrong, when you get to the end in your subtomogram averaging, you have a problem, and then you have to go back and do it again. So Ewan in my lab has written this program, and here I want to acknowledge Alistair as well, because he had a lot of input into this, and Ewan has also taken some of his scripts from before. And he's called this Tomo robot. And basically the idea here is to go from the collected data up until you know, having a series of files that are ready to go in RELION 4 in a few commands that are standardized. So this is available on GitHub if anybody is interested. And so yeah, the idea here is that you basically just input your tilt images, and the only caveat is you have to have a certain naming convention. And then you get the tilt series stack. Then you get a star file, which is the one that RELION is going to read, that will contain all the alignment transformations for the stack, the CTF estimation, it will track the dose, etc. And then it will also give you a tomogram, which is reconstruct for you to pick particles from, which will be compatible in terms of XYZ coordinates with the RELION processing later on. So I just want to make a small note about the CTF estimation. So we use CTF find 4 in the Tomo robot. And what we noticed is that if you actually estimate the CTF on the full image, the CTF estimation at high tilt tends to go wrong. And that's probably because of carbon edges often coming in and sort of really changing the, the signal is not consistent. And so what we found is that if we apply a mask that only that if we apply a mask which is smaller as you tilt, then this mask will only contain the central part of the image, and our defocus estimations are much more accurate. So we have this inbuilt inside the tomorobot software. So for the particle picking, as I said, this is project dependent. We have a number of different tricks that we use. So a lot of people picking particles on membranes. They can exploit the membrane geometry. For example, if it's tubular, you can just pick the axis of the tube, and then the particles would be automatically picked on the tube surface. You can do the same with spherical vesicles. If you have irregular membranes, that's a little bit harder, and you usually have to manually segment the membranes. So we have implemented a method where basically we use ISONET to make the membranes more isotropic, and then we use the EMAN2 AI tools to automatically segment these membranes. Then using Chimera, we select individual membranes, and then we have some scripts that will just oversample the membrane surface and give you particle coordinates. But as I said, every project is different in terms of particle picking. And here I just want to say that for us, having multiple software is really, really important. For example, when we pick our particles, we need to use a number of different software to actually get the particle coordinates. For example, we oversample the surface of the tubes, and then we need to essentially find where the center of the actual particles on the tubes are. And for these, we did try to use the RELION 4, but because the initial coordinates are so far off from the actual real coordinates, that doesn't really work. So we need to tell the software, for example, because these particles are on the surface of the tube, we have some initial angles, but we are not sure about the in-plane rotation is plus and minus 180 degrees. And these kind of ad hoc searches, you cannot really do them with the streamlined software like RELION, but then you can use software like Dynamo or other software to do these searches. So it's important for us to be able to switch in and out of different software for these projects. Likewise, when we are aligning the outer code, we want to make sure that we are searching only in X, Y, but we're not allowing the Z axis to change, because otherwise we sort of, the alignments tend to fall back onto the inner coat. And that is also something that we can't really do with RELION, for example. So, but anyway, once we get more or less correct particle coordinates, then we can switch to RELION and then do local alignments. And I just want to show you our results for the inner coat. So this is quite new data, and I'm kind of comparing it with what we published last year. And this is the very same data set, just reprocessed in RELION 4, and we had a really, really big jump in resolution. You can see the quality of the structure is a lot better. So this is another sort of zoom in into the core of the density. So this is somewhere, I don't know, around 3.5 to 4 angstrom resolution. With different software, give us a different resolution estimate, but I have these couple of movies here to show you that the quality of the density is really, really improved a lot. We see all of the side chains, and this is consistent throughout the structure. It's not just one really good part. It's good throughout. So we're really quite happy with our new pipeline. And to get to a structure like this, it took a few weeks with this sort of automatic pipeline that I showed you. Okay, how do I go forward? Right, and so this is also just to show that even the bad parts of the structure, because in our structure we also have flexible parts, somehow with previous routines they just kind of were very streaky and anisotropic, whereas with the new routines they seem to, they are at low resolution, but the density is a lot it looks a lot better and much more interpretable. So that's yeah, so the other thing I also wanted to show you is that now we have the kind of resolution, and also we really trust our density a lot better, and we can do some modeling. And this is just to show you that, so we fit the AlphaFold 2 models, and they fit really well, but there are some differences. And one of the nice things that we can say that because we are looking at the structure in its environment, so these are membrane-bound proteins, and now here we are looking at so this is the membrane down here, quite noisy, and we're looking at the membrane interacting region of one of the subunits, and we can see that when the protein is in the membrane environment, there is this histidine, for instance, which is this loop is pulled down towards the membrane, and this is quite nice because it fits with the biochemistry, because we know that we need negatively charged phospholipids for this binding to occur. And here we can really see that the positively charged residues are pulled towards the membrane. So that's quite nice to see. Another thing that we noticed that has improved a lot using the RELION 4 pipeline is our ability to classify. So before, every time we tried to classify, we always got classification based on the missing wedge. So particles that are top views were in different classes from side views, and it was really hard to actually classify different conformations unless there was a huge difference between particles. Whereas with RELION, it just works. So here you can see like 2 classes, and yes, this one looks more noisy, but both of them really look like our structure. But then when we map back onto the tomograms, the positions of these particles, we can see that this is total Einstein from noise. So these are just totally misaligned noise particles, whereas this class here is made up of well-aligned particles. And in this case, we're lucky because our particles follow a lattice, so we can really assess which class is made up of good particles and which class is made of bad particles. But there's essentially no missing wedge bias in the classification here, which is something that is quite exciting for us. How am I doing with time? Sorry? A few minutes, okay. So the other thing that I want to mention is that in tomography, we have we don't only get the structure, we also get the positions and orientations of all the particles that contributed to the structure. So we can learn a lot by looking at that. And I'm giving you a few examples on how we can use that. So one example here is simply, we use it for cleaning up the data, or Peter mentioned yesterday, we can also use this for validation. So when we map back our particles, we can see this makes sense or this doesn't make sense. I showed you earlier the example of the classification. We can also then select data based on whether their positions and orientations make sense or not. For example, in the case of the inner coat, here we have coordinates of particles, and you can see that mostly they follow the expected lattice. And if we actually plot the neighboring positions of particles for each particle, the neighbors sort of follow a certain distribution, which is the one in the lattice. But there are some outliers. So what we can do, for example, is to create masks around the peaks where we expect particles to be, and then use this mask to select. And so in this case, we would select only the green particles and eliminate the pink particles. But these we can also use to actually select different classes of particles that have different geometrical relationships. So in this case, it's just the right relationship or the wrong relationships, but we can also use it to distinguish different geometrical relationships. I'm going to skip this slide here. And this is the example. So in our outer coat, for example, we found that these green particles here, they are sort of in this geometrical relationship compared to the vertices of these cages here in red. So when we plot, and I'm not going to go into the biology at all, but when we plot the position of the red neighbors compared to each one of the green particles, we find that yes, they follow this, there's these 4 peaks which are expected, but then we also see that there's an extra peak that we didn't expect, and we wouldn't have spotted just by looking at the structure or the data, because it's a minority of particles. And so we can use our masking approach to actually distinguish the green particles that have neighbors in these 4 positions, and the green particles that have neighbors in this strange position. And then we find that some of these green particles are actually connected to a rod on one side and to a vertex on the other side, and then we can draw various biological conclusions from it. So this is quite powerful approach to interpret subtomogram averaging data. And yes, so I'm going to stop here. Hopefully I didn't go over time. And I would like to thank all of my group. I showed you Ewan's work today. Also, Sander Jenny is now left, and Christina has joined. I would like to thank all of the collaborators. Liz and Vicky are more involved in the biology side of things. The data I showed you was actually collected by Josh together with Wim, and that's what we published earlier. And of course, thank you to Wim and his team for data collection, and also people at ISMB, Dave, Claire, and Natasha, who's here, great lab manager. And that's it.